We have today
So this was my PhD project where we wanted to understand. So there are some limitations when it comes to corals. You can't just open up a human every now and then. That would lead to the eventual death of the coral. So we do something called amplicon sequencing. One approach will be to go for metagenomics, where you sequence everything and everything. It makes one particular type of amino acid and it will activate this gene, learn from Hydra, keep working with Hydra.
today we have a special guest on genetic education podcast Raw and Real, whose work inspired other scientists to understand the impact of climate change on coral and marine life. We have today Dr. Jay Bhatia. Dr. Jay, how are you?
Hi, I'm good. Thanks for having me.
Yeah, welcome to genetic education, and it is our honor to have you on our podcast.
Yeah, thank you.
Yeah. So before moving ahead, let me just give all of you a brief introduction of Dr. Jai's scientific journey. So Dr. Jai is currently doing postdoc at the University of Düsseldorf in Germany and has already published 7 peer-review articles. And trust me, those articles are published in very good journals. Very amazing work, Dr. Jai.
Thank you.
During his PhD, he works on the fascinating relationship between Green Hydra and the algae symbiosis and its importance. So, how is everything going, Jay?
It's going good. So this was my PhD project where we wanted to understand about the how it occurs in nature, basically in corals. So there are some limitations when it comes to corals, like you cannot work with them in lab so easily. It's like if you want to study something in humans, it's not accessible always. You can't just open up a human every now and then. So you go and study those things in mice, you learn from them, and then you apply the knowledge to humans. And this is something similar that we did. We wanted to understand that in case of corals, like you people must have heard that recently there is this heat waves coming in oceans, there's global warming, the average ocean temperatures are on rise. What this causes is that it leads to a phenomenon called coral bleaching. Now, corals are basically, if you look at it, they would look like colorful stones. But in fact, they are these tiny animals. They belong to a phylum called Cnidaria. And these tiny animals, they are made up of just basically two cell layers. And in the inner cell layer, they have intracellular algae.
The
So they are
algae
like,
is living inside the cell.
yes, it's living inside the cell. So it's like, we call it endosymbiont because endo means inside and symbiont is something that is in close partnership with another organism. Now, this relation is mutualistic, meaning that they both benefit each other. So the algae does photosynthesis, it produces sugars and other organic molecules that go to the corals as the source of nutrients. And corals provide fixed nitrogen to the algae. So coral can eat food from the ocean a bit, digest it, and then it breaks it down, and then it provides amino acid in the form of amino acids to the coral, to the algae. Now, because of bleaching, this algae would leave the corals, and that would destabilize the symbiosis, and this is what's called bleaching. This
Okay.
algae leaving the coral, and that would lead to the eventual death of the coral. The recovery chances are quite less. Now, as I said, if you want to save them, you need to understand them better. People are doing a lot of field studies. People are now slowly getting successful to keep them in labs. But back when I was studying Green Hydra, it was not that feasible. So what we did is we took a similar model system, which is Green Hydra, and they look basically a bit similar. The big difference is that the corals, they make big colonies. You see as these big stones. But in case of Green Hydra, they are individuals. And they have a different type of algae in the cells, but the base principle of the symbiosis is similar, basically.
Okay.
So you learn some basic things from Green Hydra about how this symbiosis work and apply that knowledge to the corals. And what I found during my PhD was that like all other organisms, or I would say most other organisms, Green Hydra has its own microbiome, has its own community of bacteria living on the animal. And it must be well known that we also have these bacteria living in our digestive system that help us with digestion, so on and so forth. And this is quite a preserved phenomenon, I would say, that's also present in these small animals. Now, the effect of bacteria is different from what we have in ourselves. Like for
Okay.
us, it's the development of immune system, for example, or motility of our gut. But in case of these animals, these bacteria protect them against other pathogens. It can protect against, like, for example, the fungal infection. It can play a role in, again, nutrient exchange, like bacteria can provide some nutrients and in return they get some other nutrients from Hydra. So there are various functions, and we wanted to see if these bacteria have the effect on the symbiosis between Hydra and the endosymbiotic algae. And we
Okay.
call this as tripartite interaction.
Yes, because all three organism are like interconnected in the symbiotic relationship. So there might be
Exactly.
interlink between all of these three organisms to survive together.
Yes, exactly. So we do something called amplicon sequencing.
Okay.
So for that, we need to know first what all bacteria are there. And one approach will be to go for metagenomics, where you sequence everything and everything. But if you just want to know the identity of bacteria and you do not want any part of host DNA in it, we do 16S amplicon sequencing. Now, the 16S
Hmm.
gene, it's specific for bacteria. So we isolate the DNA from the whole animal, and then we target specifically 16S gene with the conserved primers. So at the end, we get only the sequences of all the 16S, different 16S that are present on this animal. And with that, we can identify every single bacteria that is present on the animals.
Okay.
We did this, and we also had the genome of Hydra, and we also had the genome of the algae. Now, from the genome of Hydra and algae, we got to know that the algae, it produces this sugar by photosynthesis. The sugar is called maltose, and this maltose goes to Hydra. And in return, the Hydra has one particular glutamine synthase gene. Basically, it makes one particular type of amino acid, and it will activate this gene only when it gets sugar from the algae. So when the algae pays the fee in terms of sugar,
Got it.
it gets back the nitrogen.
Yeah.
Now,
Okay. Okay. Okay.
yeah,
Right.
and for that, we also did some comparative transcriptomics to identify how this works. So here you get information about all the genes that are expressed in the form of mRNA. And yeah, so for these two partners, we had this information. But later we, as I said, we introduced a third partner of the bacteria. And there what we found is that if you do not have the algae, if you remove the algae, the host, the animal, gets easily infected by pathogenic bacteria. In
Hmm.
our case, it was a bacteria called Legionella. It's also considered as a human pathogen and it is quite invasive. Now, if you have some animals without algae getting infected with this pathogen in a population of Hydra, no matter if the other animals have algae or not, the Legionella will start spreading in the population. It's very infectious. It will take over the whole population and it will increase the chance that the algae leaves the animal. Now, this can act as a double-edged sword.
Yes. Yes.
On one direction, this algae is leaving the animal. But if the animal has enough algae inside, it can still survive.
Hmm.
Now, this algae that left the animals, it can be taken up by other animals that do not have the algae.
Hmm.
So this is called algal propagation.
Okay.
Now, those animals will gain this algae and it will start doing photosynthesis inside this animal. And as a result of this, this Legionella will not be able to survive.
Okay.
Now, this happens under an ideal condition. We include additional stress, environmental stress.
A very, very fine balance between the algae and the host hydra. Very fine balance. If it becomes imbalanced in either cases, there are very high chances that the hydra get the pathogenic infection.
Yes, yes,
And
exactly.
you have done genetic analysis that shows that in the presence of algae, what I understand is in the presence of algae, there are certain set of genes in the hydra that get expressed.
Yes,
That is very,
yes,
very fascinating. It is like this symbiotic relation is, we can say, occurred from very long time, and then only this can be happened. Like in the presence of
Yes.
other organism, this organism expresses its set of genes, and then this organism expresses some set of genes and mutually benefit to each
Yeah.
other. Very, very
And if
interesting.
you just look at the genomics, actually this particular algae, the chlorella algae, it has lost some genes actually that would make it able to live freely without the hydra. So
Okay.
it cannot take up the nitrogen from the environment. So it
Hmm.
has to depend on hydra for nitrogen. So it has
Definitely.
to
That's
live
why.
in the hydra. So it has a genomic complementarity.
Yes, that's why they mutually mutually get into the relationship. That's the whole
Yes,
point.
yes,
Yeah.
yes.
Okay, so
And
how this
yes.
this entire research of yours, it is on Hydra and the symbiosis of an algae. So how it is associated with coral? Like how it is solve the climate change, the impact of climate change on the coral.
So to solve it, I think it would take quite some time to make a final solution. But as I said, people are working on multiple solutions. So
But
from our
based on
study,
your
what we
research.
can, yeah. So from learn is that it would happen in corals also, that if they do not have algae, they would have higher chance of getting the infection.
Okay.
Now, this is not exclusive. The corals can get infected by the bacterial pathogens like Vibrio, for example, that even if they have the algae, but they still have the possibility to recover if they have the algae. If they do not have the algae, the death is imminent. Yeah. So,
Amazing.
yeah.
Amazing. Your work is like, it is like, we can say it's not restricted to one system, symbiotic system, but it is a model kind of understanding for other system as well. And that's what
Yeah. Yeah.
other scientists are doing, implementing your model to understand the impact of climate change on the corals, right?
Yes. Yes.
Okay,
Exactly.
so very interesting. My next question is all about, first tell me how you like go Germany from PhD to postdoc at Germany.
That was a little interesting. There were some minor regrets also of some opportunities that I had to let go. Like after I finished my master's from MS University of Baroda, during the master's actually, I went for a small duration of summer training in Pune in Agarkar Institute in the lab of Dr. Surendra Gaskarbi. He is the Hydra biologist of India, the Hydra
Okay.
biologist and the developmental biologist.
The Hydra man.
Yeah, yeah. And I worked there for 2 months and then it got me hooked on Hydra. I could not think of working on something which is other model system because it's such a fascinating organism. It has exceptional regeneration. You cut it into as many pieces, each piece will make one animal. You
Okay.
can actually break the whole animal into single cells, and if you keep these cells together, they will make the animal again.
Okay.
It's that level of regeneration that it can do. And it's really simple body plan, easy to work in lab. It just appealed me a lot. We have learned a lot from Hydra that we apply for humans, basically. So I got hooked onto Hydra, and at the end of my master's, I started applying in Germany. And I found one particular lab that was working with Hydra and microbes and how these interactions take place. And that was really interesting to me. So I applied a couple of times. I didn't get any answer. Meanwhile, I got into TIFR for my PhD. And after I joined in 2 months, I got a response from Germany that we have a position if you would like to join. So it was quite a tough choice to make, but my interest of Hydra eventually won. And I had to unfortunately leave TIFR. I mean, it's a really good institute. But I ended up here and then I started working with this Green Hydra project.
Okay.
And after I finished my PhD in 2020, I joined as postdoc. Still continuing work with Hydra,
Hmm.
and now I study how microbes affect the development of animal, which is a whole another interesting domain of study. Yeah, I'm still continuing working with Hydra. I would like to soon come back to India with my Hydra and continue my research there.
The last question we have is, what is your future plan?
So for my future plans in research, I would say that I would continue working with Hydra. As I said, I am planning to move back to India and continue my research there. And I would like to focus on interaction between the bacteria and the hydra and how it affects the development eventually. And I would like to have some collaborative work. So I have some other ideas also where I would like to learn from hydra and test the results that we gain from hydra in a higher model system, like maybe rats. Or I'm also thinking of finding a collaboration that would be able to study the similar things in on organoids, like
Hmm.
these days this stem cell that makes these organoids, it's quite in fashion, I would say. And yeah, eventually I would like to broaden my horizon, like learn from hydra, keep working with hydra, yes, but confirm, validate your risk findings in a higher model system that make it more relevant to public in general.
Interesting, interesting. Best of luck for your future, Dr. Jay.
Thank you. Thank you.
And thank you so much for joining us. We get so much insight about novel research ideas and in which direction the world is going.
Thank you for inviting me. Yeah, it was quite nice talking to you about and spreading this. It's a little bit niche topic, but it's nice to make sure that
Yeah,
people.
and for our audience, all the links for Dr. Jai's research papers and what article we wrote on his work will be provided in the description. So you can explore more about the technical side of the research and get insight about the, we can say, methodology and the progress and all of these things by reading these articles. Thank you so much, Jai. See you soon. Have
Thank
a
you.
nice day.
You too. Bye.